Thank you very much for the kind introduction, and welcome also from my side to today's webinar, in which we will see why and how we should analyze short and medium chain fatty acids, which are key metabolites to study the microbiome, diet, and the gut immune axis. My name is Barbara Ustaszewski, and I'm a scientist at the Research and Development Department at Biocrates. So what can you expect to hear in the next approximately 30 minutes? First, I will briefly introduce you to the similarities and differences between short and medium chain fatty acids. Then we will discuss the role of short and branch chain fatty acids, as well as of medium chain fatty acids in health and disease. And then I'll introduce you to our newest essay for the analysis of short and medium chain fatty acids. And in the end, we'll summarize what we've heard today. Let's start with a quick introduction to short and medium chain fatty acids. Short chain fatty acids have a chain length of 1 to 6 carbons, whereas medium chain fatty acids comprise those with 7 to 12 carbons. In the human body and other mammals, short chain fatty acids are mainly generated through the fermentation of dietary fibers by our gut bacteria, while medium chain fatty acids derive primarily from medium chain triglycerides that we take up with our diet, for example with dairy milk or different plant oils. The fatty acids of both classes are readily taken up by diffusion through the cell membranes and are rapidly metabolized in the mitochondria and activated to acetyl-CoA thioesters. When it comes to their physiological roles, both short and medium chain fatty acids are probably best known as important energy supplying fuels that can also modulate the mitochondrial energy production. But there are also central signaling molecules involved in the regulation of, for example, carbohydrate or lipid metabolism. And they control also cell death and survival. So the combined consideration of short and medium chain fatty acids can provide answers about the microbiome, the diet, and energy homeostasis. After this short overview, we will now focus on short and branch chain fatty acids and their roles in health and disease. As just mentioned, short chain fatty acids derive mainly from dietary fibers, but also dietary proteins and peptides, or microbial succinate and lactate that are fermented by the gut microbiome. The three most abundant short-chain fatty acids are acetic acid, propionic acid, and butyric acid with 2, 3, and 4 carbons. Short-chain fatty acids exert their functions mainly via 3 mechanistic pathways. They can bind to G protein-coupled receptors on the cell surface and thereby activate the immune system, modulate nervous signaling, and decrease the permeability of the blood-brain barrier. Secondly, they can inhibit histone deacetylases that are important epigenetic regulators, which impacts, of course, gene expression, but has also anti-inflammatory and immune suppressive effects. And thirdly, they are integrated in our metabolism, especially the TCA cycle and beta oxidation, to produce energy, which has a positive effect on insulin sensitivity, glucose tolerance, but also cell proliferation and differentiation. So overall, we can say that short-chain fatty acids are immune-boosting metabolites that promote metabolic health. But there are certain differences between short-chain fatty acid species and their effects at different sites of the body. Acetic acid is the most abundant short-chain fatty acid in the circulation and directly affects organs throughout the body, also other than the gut, like the lungs, white adipose tissue, or the bone marrow, for example. It can also cross the blood-brain barrier, and it reduces postprandial appetite as well as intestinal transit time via the interaction with the G protein coupled receptor GPR43. Propionic acid is primarily metabolized in the liver. It can be converted to glucose by gluconeogenesis in the intestines. And it also reduces postprandial appetite and intestinal transit time, again via binding to GPR43, but also GPR41. Butyric acid is the primary energy source for colonocytes, and it exerts its anti-inflammatory effects via the GPR109A and HDAC inhibition. So we see that depending on the receptor and the tissue, short-chain fatty acids have different systemic functions that mediate the gut immune axis, but also many other gut organ axes. But since short-chain fatty acids mainly derive from dietary precursors, diet plays a major role here, whether we stay healthy or develop a disease. In fact, short-chain fatty acids were shown to represent a causal link between the diet and a wide range of disorders. An unhealthy diet that is low in dietary fibers, like for example the Western style diet, gives rise to lower levels of short-chain fatty acids produced by the gut microbiome, which can lead in the long run to different pathophysiological conditions such as obesity, insulin resistance, or inflammation. And these can in turn result in various diseases from diabetes to cancer. And there are several studies that have shown that supplementation with short-chain fatty acids or their precursors as prebiotics, or with short-chain fatty acid producing bacteria as probiotics, can actually promote health effects, including weight reduction, reduction of fasting glucose levels, and increased insulin release. Here is a nice example in which patients with multiple sclerosis were supplemented with propionic acid. This study was led by Professor Hagikia, who is now at the University Hospital Magdeburg in Germany and was published in Cell last year. More than 300 MS patients were included in the study, and they investigated on the one hand short-term supplementation of propionic acid for 14 days followed by a regular immunotherapy, and on the other hand long-term supplementation for up to 6 years. What they found was that the patients generally exhibited significantly reduced levels of propionic acid in serum and feces, and also dysbiotic gut microbiome. And already short-term supplementation of propionic acid significantly increased functionally competent regulatory T cells, whereas Th1 and Th17 cells decreased. And long-term supplementation led to a reduced annual relapse rate, as depicted in Figure A, and a reduced risk of disease progression, as shown by the Kaplan-Meier plot in B. So the long-term supplementation of propionic acid ameliorated immunological, neurodegenerative, and several other clinical parameters, which the authors explained by the stimulation of the regulatory T cells, which makes propionic acid a potent immunomodulatory supplement to the standard treatment of multiple sclerosis. A specific subgroup of short-chain fatty acids are branched-chain fatty acids. They derive from the branched-chain amino acids that are valine, leucine, and isoleucine that are taken up by the diet and fermented by the gut microbiome to, for example, isobutyric, isovaleric, or isocaproic acid. Among other functions, these branched-chain amino acids are recognized by specialized cells in the gut epithelium, the enterochromaffin cells, upon which they release serotonin, which in turn activates sensory nerves of the enteric nervous system via synaptic-like connections, which results in increased gut motility and gut epithelial secretion. So branched-chain fatty acids are another class of metabolites specifically derived from the gut microbiome and are important mediators of the gut-brain axis. So next we will have a look at medium-chain fatty acids, their physiological roles and disease implications. As mentioned in the beginning, Medium-chain fatty acids derive mainly from medium-chain triglycerides that are contained, for example, in dairy milk or in plant oils like coconut oil or palm oil. The circulating medium-chain fatty acids like caprylic, capric, or lauric acid are probably best known to serve as energy source. They are metabolized in the mitochondria via beta oxidation, they are precursors for ketone bodies, or are used in lipogenesis. But medium-chain fatty acids are also signaling molecules that exert tissue-specific regulatory functions on the metabolism. They can interact with membrane receptors like specific G-protein coupled receptors, just like the short-chain fatty acids. But other than those, medium-chain fatty acids have rather pro-inflammatory effects, but they can also confer insulin sensitivity. They can also bind to nuclear receptors like PPA gamma, which can again increase insulin sensitivity, but also contributes to adipogenesis. And medium-chain fatty acids can also interact with second messengers like AMP, which in turn activates AMP kinase, which leads to an overall increased catabolism. So, medium-chain fatty acids contained in our diet have again a strong impact on our health status, since they can influence glucose metabolism, lipid metabolism, and hormone secretion. In certain tissues, insulin secretion and sensitivity can be increased. Beta oxidation, thermogenesis, and lipolysis can be increased. And depending on the physiological context, medium-chain fatty acids can increase appetite via the activation of the hormone ghrelin, or they can increase satiety by stimulating the secretion of leptin and cholecystokinin. A paradox has actually been observed here, that is that in anorexic or cachetic patients, supplementation with medium-chain fatty acids successfully induced appetite, while in obese individuals, this has led to an enhanced satiety or even weight loss. This might be explained either by a ghrelin resistance in the overweight population or by a greater metabolic benefit of an increased thermogenesis that overrides an increased appetite. So overall, balanced levels of medium-chain fatty acids can block the development of pathophysiological conditions and diseases like diabetes or also Alzheimer's disease. There are indeed several clinical trials that have shown beneficial effects of supplementation with medium-chain fatty acids on metabolic and neurological disorders. The supplementation provides an alternative energy source to glucose, which can help to treat metabolic disorders of cellular transport or long-chain fatty acid metabolism. The supplementation also promotes lipid metabolism and weight loss, which can be exploited in case of obesity, diabetes, or hypertriglyceridemia. And it provides also an alternative energy fuel to the brain, which was shown to ameliorate Alzheimer's disease, epilepsy, and ALS or Lou Gehrig's disease, for example. Now that we know more about the relevance and the central role of short and medium-chain fatty acids in health and disease, I'd like to introduce you to our newest assay for the analysis of short and medium-chain fatty acids and certain pre-analytical notes that we have encountered during the development. Just very recently, we have launched the short-chain fatty acid plus assay, which is an LC-MS assay that provides the analysis of short, branched, and medium-chain fatty acids for the combined readout of the microbiome, diet, and energy homeostasis. With 19 analytes, it is, to our knowledge, the most comprehensive assay for short-chain fatty acids currently on the market. It provides standardized, quality-controlled, absolute quantification and includes optimized protocols for sample preparation and normalization especially for fecal samples. As with all our products, the generated data is comparable in time and space, which makes them best suited for longitudinal studies and for pooling of datasets in larger cohorts. And the highly accurate and reproducible results are also ready to be translated into the clinics. So when we have a closer look at the short-chain fatty acid plus assay, it covers 11 short and 8 medium-chain fatty acids, including 7 branched-chain fatty acids. The assay has been validated for human feces, plasma, and serum. In case of fecal samples, an amount of 200 to 500 milligram wet weight is required for optimal sample handling, whereas we recommend 3-spot sampling, where you pool 3 aliquots of 200 to 500 milligram each from different positions of the stool sample. We'll come back to that in a few minutes. You can also send 30 microliter of fecal extract or 60 microliter of plasma or serum. The absolute quantification is based on a 7-point calibration and 3 quality control levels, one set that is specific for human feces and one specific for human plasma and serum. The analysis runtime of this 69 well plate based LC-MS-MS assay is 10 minutes per sample, and the total runtime is approximately 19 hours for a set of 75 samples. The short-chain fatty acid plus assay provides an excellent analytical performance with broad quantitation ranges for all 19 analytes for both feces as well as plasma and serum, as you can see here in the table. As to be expected, we detected much lower levels of short and medium-chain fatty acids in plasma and serum as compared to feces, since after their generation in the gut, they either remain in the intestinal content or are readily metabolized by cells. So overall, the assay typically detects 18 to 19 analytes in fecal samples and 14 to 16 in plasma and serum, depending of course on the sample storage time and conditions. The following slides deal with some pre-analytical aspects that we've encountered during the development of the assay that we'd like to share with you to ensure that you get the best data quality possible based on your samples. When we compared fresh individual plasma samples depicted as green box plots and serum samples in red that were stored at -80 for only 1 month, depicted in darker colors, with samples that we stored for 4.5 years in lighter colors, we could observe a certain trend for short and medium chain fatty acids to increase with the storage time, which was a bit more pronounced in plasma samples. In other words, it seems that serum samples might be slightly better suited after long-term storage for the analysis of short and medium chain fatty acids. But as always, it is just important not to mix plasma and serum samples for metabolomic studies and to treat samples that you want to analyze together as homogeneous as possible. We have also investigated the effect of storage temperature on the levels of short and medium chain fatty acids in plasma and serum. So here we compare plasma samples, again in green, and serum samples in red, from the same individual that were stored at -80 degrees, depicted in darker colors, and -20 degrees in lighter colors. And you can clearly see that the concentration of short and medium chain fatty acids not only differ between plasma and serum, but increase with storage temperature, which might be due to ongoing degradation at higher temperatures. Unfortunately, such stability data are not available for feces yet, but we would expect to see certain instabilities there too. But the most important thing is, of course, as always, that all samples in a sample set were treated and stored the same way so that the obtained results are not affected by such instability issues. In the case of fecal samples, we have specifically investigated the sample collection since feces is a highly heterogeneous sample matrix. As mentioned before, we have compared one-spot sampling, shown in the upper panel, to three-spot sampling, shown in the lower panel, where 3 aliquots of 200 to 500 milligram each are pooled. In both protocols, we homogenize the sample with 3 times the amount of isopropanol and then centrifuge it and take a 10 microliter fecal extract for the analysis. Due to the larger sample volume in the case of the 3-spot sampling, an extra vortexing step is required to generate a pre-homogenized pool from which an aliquot is taken for proper homogenization. So here you see the direct comparison of these two sampling approaches, and you can clearly see lower coefficient of variance values of less than 15% in all 15 individual fecal samples tested by the 3-spot sampling method on the left side, as compared to one-spot sampling on the right side. When 3 aliquots of a fecal sample were analyzed individually, the CVs were much higher, which reflects the heterogeneous nature of stool samples. So based on these findings, we highly recommend the 3-spot sampling approach whenever applicable. We have also compiled all these pre-analytical aspects in an application note that you can download from our homepage. And with this, I'd like to summarize what we've heard today. So first, we have seen that short and branched chain fatty acids are microbiome metabolites with both signaling and immune boosting roles that promote metabolic health. We then have learned that medium chain fatty acids are a diet-derived energy source and exert tissue-specific metabolic regulatory functions. And I have introduced you to the new short chain fatty acid plus assay that provides standardized quality-controlled absolute quantification for the broadest coverage of 19 analytes, 11 short and 8 medium chain fatty acids. It allows the analysis of plasma, serum and fecal samples, for which we have developed optimized sample handling protocols to minimize pre-analytical issues that are all available as application note. So in conclusion, the combined analysis of short, branched and medium chain fatty acids provides a more complete picture of the microbiome and energy homeostasis. You can find more information on the short chain fatty acid plus assay, including the application note, on our product page, or you can read our blog article to get more background information on short and medium chain fatty acids. Please feel free to contact me directly for any further information or in case you have any literature requests. And yeah, with this, I'd like to thank you very much for your attention, and I'm happy to take your questions now.